Good morning, everyone. Welcome to the webinar for February. My name is Rebecca Bailey, and I will be your moderator for today's webinar. Our speaker today is Dr. Brian Topp. Brian will be taking questions at the end of the webinar. You can ask questions at any time during the webinar by entering them into the questions box on your GoToWebinar side panel. Brian Topp is a Senior Director of Systems Pharmacology at Merck. He has spent the last 8 years developing and applying systems pharmacology models to the oncology pipeline at Merck. Prior to that, Brian led systems pharmacology groups at Eli Lilly and Intelos. This work focused on the prioritization and optimization of therapeutic strategies for the treatment of type 2 diabetes. Brian received his PhD in kinesiology at Simon Fraser University in Burnaby, British Columbia, Canada. And I would like to give a very warm welcome to Dr. Brian Topp.
Good afternoon, everybody. First, I'd like to thank you all for taking your time to listen to my talk today. Secondly, I'd like to thank Christina and Rosa for inviting me to speak at this very prestigious forum today. I'm going to break my talk today into two pieces. I'm going to start by talking about the role of systems pharmacology in drug discovery. That section's called From Discovery to Design. And then I'm going to talk a little bit about some systems pharmacology work that me and some others here at Merck have been working on in looking at lesion-to-lesion and patient-to-patient heterogeneity in cancer. So the second part of my talk will be called Wargaming Our Fight on Cancer. Have you ever noticed that Lockheed Martin doesn't discover fighter jets? Or that Apple doesn't discover iPhones, right? Ferrari doesn't discover F1 race cars. In fact, I can't think of a single other research industry that uses the word discover to explain their R&D and advancement in technology. So why is it that we refer to drug discovery, to discovering drugs, instead of engineering or building drugs? I guess this comes down to the first law of predictive simulation. If you don't know how your system works, you're unlikely to be able to predict how it's going to respond to a novel perturbation. And I think back in the 1950s and prior to that, this was almost completely true. We had limited insight and limited knowledge of how our system works. So a model-based prediction of the system was unlikely to generate highly accurate outcomes. But I'd like to ask you, do you really think that's true today? Right? How much has our knowledge changed? And how much is it changing over the next 10 to 20 years? Have we reached the inflection point where we switch from discovery to design? Or as I would argue, I think we reached that point probably 20 years ago. So what is it that makes biology so difficult to model, so difficult to predict? I would argue that it comes down to two basic things. On the left, we have complexity. This picture, though it's hard to see, is a map of metabolism inside of a cell. And you can see that there are many, many, many metabolic intermediates. There are many pathways, and there are many complex feedback loops, both positive and negative, and most of these are nonlinear. So we are dealing with an incredibly complex system. On the right, what we have is the other thing is uncertainty. While we have mapped some of human physiology, there are still huge gaps left to be explained. So here I found an early map. As you can see, there are parts of this world map missing, like, North America. But the other thing I found interesting on this map is this was the first map ever to use the phrase "here be dragons." And it reminds us that in human physiology, some of the things that we think are.
Hi, everyone. I'm sorry. We are having a bit of audio issues. And so I am interrupting. We're going to try and get the audio issues corrected. Brian?
Yes. Would you like me just to present from my computer?
Yes, please.
All right, here, let me get this going. Can everyone see my screen?
Yes.
Ah, perfect. Okay, so let's see. I thank everyone. Discovery to design.
It's showing the desktop, Brian.
Showing the desktop.
Yes.
Okay, how do I change that?
Are you presenting your slides?
I am.
It's probably showing, you probably have a dual screen and it's showing.
Yeah, show monitor 2.
Yes.
Can you see my slides now?
Yes.
Fantastic. All right, I apologize for the little glitch. A nice picture of a fighter jet though, huh? All right, so where were we? Yeah, so here you look at, why is predicting clinical outcomes in clinical pharmacology, in drug discovery, why is that so hard, right? So we've mentioned on the left, the system's very complex, non-linear, multiple feedback loops. And if you look at the right, this map of the world, right, there are things missing, obviously, like North America. And then we have this thing written here, here be dragons, which, as you move forward in time, it turns out not to be true. There were actually no dragons there. So we have things we believe to be true that in the future will turn out not to be true, and there are just pieces of information missing. So in the early stages, where we had a limited understanding of what we were trying to discover, what we were trying to accomplish, it truly was a world of of discovery. We would go down to the Amazon and find some exotic beetle, crush up its shell, and ask, will this beetle cure cancer? Right? We'd go to Africa and we'd find the bark of a tree that the tribe would ancestrally chew to prevent disease, and let's see if that works, right? And eventually, we took this to an extreme level with high throughput systems where we would create thousands and thousands of chemicals, and we put them on thousands and thousands of cells and just hope that we would accidentally create a molecule that happened to cure cancer, right? I mean, it kind of reminds me of the story of putting 1,000 monkeys and 1,000 typewriters and hoping that one of them writes War and Peace, right? Now, stage 2, as we were doing all this discovery in the background, people started moving towards rational target selection, right? We wanted to know, if we understood how the system worked, we would probably be better off at predicting how it would respond to to novel perturbations, right? If you want to cure a disease, you have to understand it. And a lot of academics and some in industry started collecting these pathway maps and these understandings of what separates normal from the natural history of disease. What are the key players? What are the things we're trying to accomplish? Are there indirect ways we can accomplish these things? And we really started to accumulate sort of a deep knowledge of how physiologic systems work. But the challenge at this stage of the game was that most of this work was done most of this work was done with thought models. People would read a lot of papers, they'd develop an understanding of how the system worked, and they would run mental simulations. And it's very difficult to run complex integrative simulations in your head. And everybody had a different model of type 2, of type 2 diabetes or cancer or whatever, because everybody read different papers and they came from different backgrounds. So there was a lack of transparency. When someone told me, I think, this drug's going to work, I didn't necessarily know what data he was basing that presumption upon, right? And when people made predictions, they weren't quantitative and integrative, um, predictions. They didn't say, well, I think A1C is going to drop 1.23%. They'll just say this target seems like a reasonable target, right? And they wouldn't extrapolate out to say, well, when glucose goes down, insulin will go down, and then free fatty acids will go up, and those high free fatty acids will cause a problem. They just sort of looked at the feedback loops within the liver and said, within the liver, this seems like it's a good idea. So as we moved on to stage 3, what we did, what we started doing, this is about the year 2000, is we started building quantitative, integrative mathematical models of human pathophysiology, right? And we wanted to know on that chart of knowledge versus predictiveness, had we accumulated enough knowledge that our predictions would be reasonable or worth integrating into a decision, a decision matrix in a pharmaceutical company. Now, the process here is pretty straightforward. The first thing you do is you build a mathematical model of the human pathophysiology, right? This is how we think the system works, right? The second thing you do is you build a model of the human pharmacology. You say, this is what we think our drug does, right? Now, you can put those two models together and simulate it and say, based on our present knowledge of physiology and pharmacology, this is what we expect to see in the clinic. And the last thing that you try to do is you try and estimate confidence in this outcome. If this simulation was premised upon a high volume of high quality human data in a system that's pretty well characterized, there's reason to believe that these simulations, are somewhat accurate. But if you were trying to extrapolate into an area that's poorly understood, you had limited data, you filled the model full of assumptions and data from a petri dish and a mouse model, well, maybe we're a little less confident in this, right? You could also do a lot of like sensitivity analysis, where you could run a simulation based on your assumption, and then you could run another simulation based on someone else's assumption. And if that uncertainty has little to no impact on your outcome, then you're fine. But if that uncertainty has a huge impact on whether your drug's going to work or fail, one, you're less confident in your model outcomes, and two, maybe that's a piece of data you have to start collecting. So we decided we were going to build these models and we were going to start making predictions about drugs, and we would see how we did, right? So, as a young PhD student, full of hair and a bright red beard, both of which I miss, I came into Entellus and I was assigned a glycogen phosphorylase inhibitor, right? So, the question is, will this be an effective treatment for type 2 diabetes, right? So we went about and we built a model and we simulated outcomes. And the model predicted that there was going to be failure, right? So if you stop the breakdown of glycogen, but you still make glycogen, the glycogen will accumulate and accumulate, and at some point that glycogen saturates. You just can't make any more glycogen. At that point, you stop synthesizing glucose. And when you stop synthesizing glucose, your efficacy kind of goes away. Also, glycogen phosphorylase was an enzyme that insulin kind of shuts off about 12 to 16 hours a day. So, when we ran these simulations, we predicted you would see, a good drop in blood glucose levels, but only for a short period of the day, and that effect was transient. It would only last a few weeks, right? So we took these simulation results and we brought them to our customer, and we explained to them what we saw and why we saw it. And of course, the feedback was, you're an idiot, your model's wrong, please stop wasting our time. And I mean, what do you expect? This was our first ever prediction. All of their experts, internal and external, had told them this was a great target, right? So we apologized profusely and we backed out of the room. But the weird thing was we were right. This target failed for the exact reason that we said it was going to fail. right? And amazingly, as we moved on through other targets, glycogen phosphorylase, fructose bisphosphatase, glucagon receptor antagonists, glucose kinase activators, SGLT1s, GIPs, MTPs, we predicted failure for all of these drugs, right? And there were dozens of them. And each and every time we were told, you're an idiot, your model's wrong, stop wasting our time. But each and every time we turned out to be right. Now, we didn't just predict failure, right? Because, almost every drug fails. 9 out of 10 drugs fail. You can just predict failure, and you're right 90% of the time. We were also able to predict and identify the 3 drugs that transformed diabetes care in the early 2000s, right? We found that inhibiting DPP-4 would cause, an increase in insulin levels in a glucose-sensitive way. You would get efficacy, but the most important thing is you would not get adverse events. The efficacy would be less than you see with the sulfonylurea, but the risk of hypoglycemia, mild, moderate, severe, or even death was basically eliminated by these DPP-4 inhibitors. Right. And our model, picked that up, and we recommended that this was a drug worth highly investing in. I wrote SGLT1 here, but that's really an SGLT2. That's a quite interesting drug that it has nothing to do with the pathophysiology of diabetes, but you find that if you just make the kidney remove glucose from the blood, you can actually get better glucose control. Now, this one was a little difficult in that it was contingent upon would the patients increase their food intake, right? If you lose glucose out the kidney and then you eat an extra hamburger, you didn't really get anywhere. And it's really difficult to predict food intake, right? So, we kind of have to put an asterisk on this one. We said if their food intake remains reasonable, it will be a diabetes target. But, so I think that's about as good a prediction as you could make. And of course, the GLP analogs, the short and the long acting, we predicted the difference between the 2 of them. And we predicted that both of those would be worth investing in. Now, there were some targets that we just simply weren't willing to pursue, right? People would come to us and say, can you help us predict PYY effects on food intake and thus glucose control? And I'd ask them, what does PYY do? And they would say, I don't know. And if you don't know what PYY does, it's awfully hard to model. So we had a really good scorecard. We made a lot of accurate predictions. But you have to keep in mind that there were a few that we didn't get right. And there were a bunch that we passed on, right? We were able to pick and choose. I will model this one because I know there's a lot of data there, right? And I'm going to skip that one because that's not an easy one to predict, right? But, overall, if you ask the question, oh no, I hit the wrong button. If you ask the question, I got a lot of slides here. Apologies. All right. Almost there. There we go. That looks familiar. If you ask the question, had we moved far enough up that knowledge chain where a mathematical model could make reasonable predictions of clinical outcomes for novel drugs, I think these early studies clearly showed that the answer to that was yes. And what we really need is academic publications that show what is our scorecard. How often do we get it right? How often do we get it wrong? Where are the models accurate? Where are the models struggling? And having that kind of transparency would really help people understand, not only what's in the models, but really how good are they, right? Now, if we look to the future, right now, we take existing targets, we put them in a model, and we simulate out a prediction. Will our existing model work or fail? Or will this drug we have in our pipeline succeed or fail? But you really want to ask the question, what should be in our pipeline, right? And if I have an ADC, how do I make my ADC best in class, right? If I have, how do I make my drug the best it can possibly be? If you're going to design an insulin, and you can build any insulin, what would you build? Would it be fast? Would it be slow? How slow? Would it be specific to the liver, specific to adipose tissue, right? Would it have fast K on, K off rates? If you can design any insulin, what would you build, right? And we started, when I was working at Eli Lilly, we started designing insulin analogs. We started asking the questions, how fast should a fast insulin analog be? How slow should a slow insulin analog be? And then we started moving on to the next steps of asking the question, what targets should we have, or what combinations should we have? And I think the next stage, right, if you think back to when Deep Blue beat Kasparov at chess, we need to imagine a world where Deep Teal, right, Merck's Teal, Deep Teal beats cancer with the personalized medicine drug. I want to build a model of cancer pathophysiology and its deep heterogeneity and its complexity. And I want to ask an artificial intelligence engine to beat that model. Go in, manipulate that model, look at monotherapies, combination therapies, personalized strategies, find a way to beat the model. All right, so if you want to have a model that beats cancer, it's not just going to happen. There are a few basic steps. The first thing you have to do is you have to collect the pertinent human data, right? I mean, when we run clinical trials, we generally say, thumbs up, it worked, or thumbs down, it failed. But every time a trial fails, we need to take that data and integrate it into a mathematical model, understand mechanistically, why did that fail? We thought it was going to work, it didn't work, what did we learn? Right? And every time we go into the clinic, we need to collect pathophysiology data and not see that as an expense, but see that as an investment in future predictive capacity. The second thing you have to do, you can collect all this data, you can't just remember it all, and you can't integrate it in your head. The system's too complicated for that. So you need to take that data and integrate it into a model of cancer pathophysiology. Now, once you have this model, you have to train and test this model on all of your past and present studies, right? This model, you should see how many of the past drugs it predicts accurately. Every time you run a drug into the clinic, you should predict the outcome before you go in the clinic and test how accurate the model was. And with this integrative loop, as time moves forward, your model gets better and better and better. And I mean, imagine a world where Merck understood the pathophysiology of cancer better than our competition. Rather than having the same pipeline as everyone else, we could leverage our inside knowledge and leverage our artificial intelligence to develop strategies that don't exist in other pipelines. Now, if we move on to the second part of the talk, wargaming our fight on cancer, so I was a diabetes researcher most of my life, and about 9 or 10 years ago, I switched over to cancer, maybe 8 years. And, being a diabetes guy, I didn't know much about cancer, and I thought what success would look like would be this patient over on the left, right? This patient has 4 tumors, and all 4 of those tumors shrink. Our drug works. Isn't that wonderful, right? And failure would be what we see over on the right. The patient has 4 tumors, all 4 tumors grow. Isn't that terrible? Our drug didn't do anything. But as it turns out, when you look at the RECIST scores for how we actually calculate success and failure, and you look at what data looks like in the real world, it turns out that most patients look like this. They have 3 tumors growing and 1 shrinking, or 2 growing and 2 shrinking, 3 shrinking, 1 growing, or you can have every tumor shrinks, but a new metastatic lesion appears, and that's failure. So according to RECIST, so first off, this is what the real world looks like. You never see all shrinking or all growing. Those are quite rare. The heterogeneity, the mixture, some shrinking, some growing, that's what you see in almost every trial. And then, according to our RECIST criteria, all of this is failure, right? But is it, right? I'm going to ask you today that question. Is this drug failure, right? If every lesion shrank, but a small new metastatic lesion appeared, it appears for 6 weeks and then it disappears. and we say that our drug didn't work in that patient, if 2 of our lesions shrank and disappeared, our drug clearly did something, right? Is this a reasonable definition of failure? And if we're making clinical decisions based on a flawed definition of what's success and what's failure, how's that impacting the quality of our decisions? So I'm going to show you an average patient. This is from Keynote-01 metastatic melanoma. This patient comes into the trial with something like 20 tumors. And we put him on pembrolizumab, and about a third of his tumors grow, a third of the tumors stabilize, and a third of the tumors shrink, right? And of course, RECIST declares this progressive disease. This patient has failed, right? But when I look at this from the point of view of efficacy, 2/3 of the lesions were either stabilized or shrinking. Like, is this really drug failure? It's not like pembrolizumab didn't do anything. It shrank 2 out of every 3 lesions, right? And the second question I asked is, should we really discontinue pembrolizumab? Like, I know this patient wasn't cured, right? Those growing lesions are going to continue to grow and metastasize, and they're going to eventually be lethal. But if we take away pembrolizumab, those 2/3 of the lesions that were responding are now going to start growing. So do we connect drug failure to drug discontinuation? Right now we do. Is that something we should continue? Now, the third thing that I thought about was, what does this mean for biomarker data? If I randomly sample one of those lesions and look at the biomarker data, what does it tell me? I mean, right now, we don't know which lesion we sampled. We just say one of these lesions were sampled and it had a lot of T cells. Does that mean, like, you know, was that taken from a growing lesion? Was it taken from a shrinking lesion? Does that mean that you can have lots of T cells, but the tumor will grow anyways, right? Or, you know, it's just impossible to interpret our biomarker data if you don't know if you took it from a lesion that grew or shrank. The fourth thing that I asked, the fourth and last thing, is how does this impact our therapeutic strategies? Like, each tumor has about 1 billion cells, and they replicate once a month, right? And you have multiple tumors, 10, 15 tumors spread all over the body. Each time you have a replication, you get a mutation, right? 1 in 1,000 mutations is a meaningful mutation. These cells are different. Genetically, they're different. Epigenetically, they're different. Their response to therapy is different. If you have 10 lesions, you'll get 10 different responses to therapy. Do we really think we're going to find a monotherapy that's going to address all 10 of those lesions, right? And then this one patient with 10 lesions is different from a second melanoma patient with 10 lesions. And melanoma isn't small cell lung cancer, and it's certainly not pancreatic cancer. So our approach of looking at one drug at a time to address a highly complex system is probably a flawed strategy, right? There is no way we're going to address this without combinations, doubles, triples, and probably personalized combinations, right? But when you try to, you know, if you go to discovery and say, I'd like to develop a personalized combination of 6 different antibody drug conjugates or, you know, checkpoint inhibitors, I mean, the laughter won't stop for weeks. You cannot develop more than one drug at a time, right? You have to show that each drug is safe, each drug is effective, each drug is dosed at the right level, that the patients would, you know, benefit more from the combo than the mono, than either mono or the control. Just our systems aren't set up to address the problem that we're trying to address. So if we move on to the agenda for this part of the talk, the first thing I want to do is just say, look, I've told you that heterogeneity is all over the place. Well, let's just characterize that. How much heterogeneity is there? Is it more in one indication than another? The second thing that I've told you is that at the time of progressive disease, more than half of the lesions are stable or shrinking. And I've suggested that maybe these patients can stay on pembrolizumab beyond the point of progression. So what we need to understand is, if we treat these patients beyond progression, will those tumors remain small? Will they remain stable? Or will they at some point rebound? Is there any benefit to continuing to treat beyond the time point of progression, right? And then I'm going to talk through some disadvantages of using RECIST as a decision-making tool. So if we start by asking, how prevalent is this, right? So I'm going to start with an example of Keynote-01 metastatic melanoma, right? This was our first ever pembrolizumab trial. Over on the right, we have a progression-free survival graph. There were 511 patients in the study who qualified for this analysis. You had to have 2 scans, one at baseline, one on therapy. And 313 of those patients developed progressive disease. Now, we're going to split this progressive disease into 2 categories. 200 of these patients have what we call primary PD. These are the worst of the worst. They develop progressive disease at the very first scan. Your very first doctor visit, you come in and your doctor says you have progressed. The drug did not work at all. And then there were 113 patients that have what you call secondary progression, where their first visit, second visit, third visit, you're doing well, but by your 4th or 5th visit, you've rebounded and you have now progressed. So we're going to look at both of those groups and see how heterogeneous they are. Primary. All right, so now if we start by looking at primary progression, over on the left, we have a waterfall plot. So the bars, the solid bars there show you the average, right? The sum of longest diameters for, if you have 10 tumors, you add them all up and you look at the average change in those 10 tumors. Now, if these patients dropped out at their first doctor visit, what you expect is they dropped out because their tumors grew, right? So you would expect to see at least a 20% increase in their sum of longest diameters. And that turns out to be true for about half of the patients. But the other half of the patients show either stable or shrinking tumor burden. So half of the patients that we declared failure actually have stable or reduced tumor burden, right? And again, these are the worst of the worst, right? And now if you look at the individual dots and those red lines, these are the individual tumors, right? So patient 100 has 7 tumors, so he gets 7 dots on this graph. The lesion-to-lesion variability is enormous. It is absolutely enormous, right? A patient with no change in tumor burden on average will have one tumor that shrinks by 60%, another tumor that grows by 80%, and 3 or 4 tumors somewhere in between. Now, the individual outcomes are just incredibly variable. And if you move over to look at the graph on the right, we ask what percentage of lesions are actually progressing. And over on sort of the top left, we have a few patients where Pembro just didn't do anything. Every lesion grew. Surprisingly, over on the right, we have a reasonable number of patients where every lesion shrank, right? Every lesion shrank, but they got PD because of a non-target lesion or the appearance of a new metastatic lesion, right? It's hard to imagine that you get 100% reduction, a complete elimination of all pre-existing tumors. We call you failure and remove you from the trial. Now, if you look at this graph, you see on average, the median patient, right, patient number 100, has about 50% of their lesions growing and 50% of their lesions shrinking. So on average, patients with PD have half of their lesions growing, half of their lesions shrinking, and that spread with some patients doing worse, some patients doing better. Now, if we move on to look at secondary progression, now with secondary progression, the assumption is the tumor burden shrinks and shrinks and shrinks, and then at some point it turns a corner and it starts growing again, right? So we wanted to ask the question, how much rebound do we actually see? And now we have a waterfall plot on the left there where basically we're showing the change from the minimum size, right? So you'll go from the minimum to the size at the time of PD. And only about 1/3 of patients actually show rebound, actually show tumor growth. The other 2/3 of patients develop progressive disease because the appearance of a new metastatic lesion. And if you ask how many tumors rebounded, the graph on the right shows you that only 29% of tumors actually rebounded. So that means at the time of progression, right, 70% of the lesions are stable or shrinking, right? There's only 30% of lesions that actually grow in patients that we call secondary progression. And that was metastatic melanoma. What do we see in other indications? We looked at multiple indications. Here I'm going to show you melanoma, non-small cell lung cancer, and gastric cancer. And even though these cancers are dramatically different, right? Melanoma responds well, non-small cell lung responds then rebounds, and gastric just doesn't respond much at all. These waterfall plots and the percentage of lesions growing look remarkably similar across these indications. About half of the lesions are growing and about half of the lesions are shrinking. So the take-home message from this point is that most patients treated with pembrolizumab show clear efficacy in a meaningful subset of their lesions at the time of disease progression. Now, if we move on to look at the post-progression period, right, what we have is we have studies where patients are given pembrolizumab, they develop progressive disease. And then the doctors call us and they say, look, I know the patient has PD, but he's doing really well. He's got more energy, he looks good, right? He or she is doing really well. Can I keep the patient on pembrolizumab, even though they're no longer in the study? And you say, yes, absolutely. Keep them on pembrolizumab and let's see what happens. So we continue to take scans to see in this subset of patients. Now, keep in mind, this is not a randomized control study. Like I just said, the patients that are doing well, their doctors want to keep them on pembrolizumab. Some patients who are just so close to the end that there's no point changing them to something else. But most of the patients in this group are patients who are doing pretty well and their doctors wanted to keep them on in the post-progression period, right? So we take a look at the tumor dynamics in these groups. And now this slide's going to take me a couple minutes, but I think it's worth it. So you would think that if you had a growing tumor at the time of progressive disease, in the post-progression space, the tumor would continue to grow, right? And we saw that in 6 patients, right? In another 24 patients, we saw tumors that were growing at the time of PD, turned a corner and stabilized. And then surprisingly, we actually saw in 7 patients, they saw growth, they were declared PD, and then their tumors began to shrink. And keep in mind that our definition of PD is confirmed PD. So we have at least 2 scans that show growth, and then at a 3rd or 4th time point, the tumors start to shrink. Right. Now, if you move on to look at patients who have stable tumor dynamics at the time of PD, you would expect if they were stable at the time of PD, they would remain stable. And mostly that's what we saw. In a small subset of patients, we saw a rebound where the tumor started to grow. And in a, my computer's locking up. And in a small set of patients, and actually not that small a subset of patients, right? 21, we saw their tumor dynamics switched from stable to shrinking in the post-progression space. And if you move on to look at patients whose tumor were shrinking at the time of PD, you would expect it to continue to shrink. We did in fact see that, but most patients transitioned from shrinking to stable tumor dynamics, and only a small number of patients continued to grow. So the take-home message from this is a shocking 24% of patients showed a meaningful tumor reduction, a 30% reduction in their tumor mass in the post-progression period. 65%, right, an enormous percentage of these patients showed stabilization of tumor burden in the post-progression space. And only 11% of patients showed a worsening of tumor burden in the post-progressive space. So here you can see this kind of repeats the point on the left, that's the waterfall plot. But if you look at the right, this is a swim plot, which tells you how long patients remained on trial. And there are a few patients at the bottom here who stayed on the trial 12, 16 weeks and then dropped out. But if you go to the top of the graph, we have patients that were declared progressive disease at week 12 and remained on Keytruda greater than 5 years, right? A lot of these patients were on post-progressive treatment for years and years. Right. Now, the last thing I want to touch on in this section is new metastatic lesions, right? So the absolute #1 cause of progressive disease is the appearance of a new metastatic lesion, right? And one would assume the graph on the left, a new lesion appears, and then it just grows and grows and grows because that lesion doesn't respond to therapy. But you can see here that out of our 86 patients, only 28 of them, when a lesion showed up, did it continue to grow. In 44 of the patients, a new lesion would show up and then it would stabilize. And in 14 patients, the new lesion was transient. It would show up, stick around a while, and then it would completely disappear. So declaring patients failure at the time of appearance of PD, doesn't appear to be justified by these numbers. So the take-home message from this point is that most patients treated with pembrolizumab show a clear and continuing efficacy at the time of progressive disease, right? That the pembrolizumab has had an effect on a large percent of the lesions, and continued treatment will result in continued efficacy. So now let's move on to our criticism of RECIST and see if we can do better here. So in early clinical trials, the primary endpoint is the objective response rate. And this is basically the best response observed during the trial, right? And, so the first question I have is good really good, right? When we get an objective response, does that really mean we've benefited this patient, right? And if you look at patient #1, the tumor burden goes down and it stays down. The best response is a partial response. And it really looks like our drug extended overall survival in this patient. You have every reason to believe that the drug's doing something good. If you move on to patient #2, we see a reduction in the tumor burden and then an immediate rebound, right? We see this a lot in targeted therapy and chemotherapy. And I'm not sure if a 12-week transient in tumor burden is really going to translate into long-term survival. So we declared this patient a winner. We high-five and say our drug's doing fantastic, but in actuality, it really only gave the patient like 6 or 12 weeks of reduced tumor burden. And oftentimes it kills sort of the weak tumors and it leaves the aggressive tumors behind. And the growth rates of the aggressive tumors are much faster than the tumor was growing before therapy, right? And if we move on to patient #3, again, we see this a lot in chemotherapy. The tumor burden goes down, the patient gets labeled a partial responder, then we have an adverse event and the patient drops out of the trial. Well, again, there's no reason to believe that this drug is going to have a meaningful impact on the patient's life, right? So there is no intellectual congruity. We see many partial responders that really aren't expected to show a good overall survival. And then we ask the question, how bad is bad, right? If you're a progressive disease, stable disease, if you're not a responder, does that mean that you're really not going to benefit from this drug? And if you look at the graph on the left, every tumor grows, pembrolizumab didn't do anything. No, you're absolutely right. PD means this patient's not going to survive long. But if you look at the second patient, right, two tumors are growing, a target and a non-target lesion, but two tumors are stable or shrinking. Pembrolizumab may in fact benefit this patient, or more specifically, pembrolizumab might be a useful part of a combination therapy that could really help this patient survive a long time. And if we look at patient #3, as I showed you in the previous section on post-progression, we have a good number of patients that get declared progressive disease at week 12 because of a new metastatic lesion, and they stay on the trial for 5 years. But we tell ourselves and the FDA and our stockholders that our drug didn't work in this patient, right? And it just isn't true, right? So our markers of failure really aren't intellectually consistent either. Now, it's one thing to throw stones and say RECIST doesn't work. Do I have an alternative? We've started to look at this. I have no proof yet that it's going to be a better predictor of overall survival, but intellectually it makes sense to me that this type of approach will work. What we need to ask, rather than looking for correlations, right? You know, marker A correlates with OS, marker B correlates better with OS. We need to mechanistically understand, did our drug do what we asked it to do, right? That's how you define efficacy. Did it do what we wanted it to do? So what do we want our drug to do? All right, most of our drugs are designed to kill tumors. So we want to say, did the tumor shrink? So we need to measure how much did our drug shrink the tumors. No point shrinking the tumor for a day or two only to watch it rebound. So question 2 is, how long did it stay down? And you're going to have to multiply those two things together to get an area under the curve, right? How deep and how long, and that'll tell you how well your drug's working. And I think a lot of people have talked about this in the literature, but what people have vastly ignored is sort of lesion-to-lesion heterogeneity, right? If you shrink 2 lesions and 2 more lesions grow, those growing lesions will eventually grow, metastasize, and they'll be fatal, right? If you shrink 9 out of 10 lesions, that 10th lesion is in all likelihood still going to become, terminal. When you go from a monotherapy, if your monotherapy shrinks 50% of the tumors, you want your combination to shrink 75% of the tumors. If you create a combination therapy that hits the same 50% of tumors that the original monotherapy hit, I'm not sure you've accomplished anything, right? So we need to start thinking about what percentage of tumors are responding to our drugs or our combinations. Next, if we look at lesion level discontinuation criteria, right, I think this is pretty straightforward. I'll just give an example. If you look at the patient on the left, he has 1 tumor, it's growing, no reason to believe your drug works, move him on to something else. The patient on the right, you have 1 lesion, it's shrinking, well, that's fantastic, keep him on pembrolizumab. But in the real world, when we get these patients that have a mixture, we usually say, oh, this patient has failed, let's take away their drug. I would rather look at this at a lesion level and say that this lesion needs a new drug. It's not responding to pembro, it needs a new therapy. But this lesion is responding to pembro. There is no justification for taking pembrolizumab away from this lesion. If we move on to incorporating heterogeneity into our biomarker stuff, I'm going to start by showing you 2 different patients, right? The patient on the left and the patient on the right are identical. They each have 1 growing lesion and 1 shrinking lesion. If we randomly sample the growing lesion, this patient is called a cold non-responder. If we randomly sample the shrinking lesion, the patient is called a hot non-responder. They are in fact the same patient. I do not see how we can categorize patients into different categories based on which lesion we randomly sampled. And I think more worrying about this is what we do with these samples is we'll sample a lesion from a patient who failed, which is both of these patients, and analyze that data to try and figure out, why did my drug fail? And if you're looking at the data from the patient on the left, you're probably going to learn something. But if you look at the data from the patient on the right, you're looking at a tumor that shrank. It did exactly what you wanted it to do. And then you're looking at that data to find out why your drug didn't work. I don't think the answer is in there. And in fact, I think we look at data from hot lesions and we develop drugs that are correlates, right, that are based on markers that correlate to good response, right? Anyways, I shall move on. Now, just one last, this is an old argument and it's a little tired, but I like to make it anyways, right? That I think the pharmaceutical industry is too committed to correlative analysis, right? We like to get big data. We like to look for statistical correlations. We get AI to correlate everything and look for what there correlates with OS, what correlates with the time of dropout. But, you know, as we all know, correlation is not causation, right? And there's a really good correlation between the sun coming up and a rooster crowing. But if you ask the question, hey, maybe if we killed that rooster, the sun wouldn't come up, that's probably not a great question. But I would argue that the pharmaceutical industry kills a lot of roosters. We look for correlation instead of causation. Instead of building mechanistic models and predicting outcomes, we do large-scale statistical analyses and chase down correlates. And even these correlates work a lot in vitro, but when you move on to integrated systems, they tend to struggle. The last thing that I want to say, and I know I've gone a little long here, is that I've shown you heterogeneity in response to pembrolizumab, right? So some patients are cured. Every lesion is PD-L1 positive, every lesion is hot, and they all shrink, and the patient is cured. But most patients show a transient response, right? So the PD-L1 positive lesions shrink, the negative lesions grow, the patients show transient benefit while the hot lesions shrink, and then they show a negative effects as the cold lesions grow. It's clear to me that these patients need some sort of combination to address the hot and the cold lesions, right? And if we move on to look at chemotherapy, we see the exact same thing, right? Some tumors are sensitive to the chemotherapy, and some patients are cured by chemotherapy, but most patients have some tumors or some subset of tumors that respond, that are sensitive to the chemotherapy. And then they have tumors that are resistant to the chemotherapy, and the majority of patients show transient benefit, rebound, and then failure. And if you look at targeted therapy, right, some tumors are HER2 positive, right, some tumors are BRCA positive, some tumors are HER2 negative, some tumors are, BRCA negative. And we see the exact same thing. A small subset of patients are HER2 positive in every cell in every tumor, and they're cured, but most patients show a transient response. And now that the pharmaceutical world's obsessed with antibody drug conjugates, what do we think we're going to see there? I mean, I would argue that basically we're going to see the same thing that we've seen with every other therapy, right? Some patients are going to have, they're going to be TROP2 positive on every tumor, right, in every organ. But the majority of patients, and again, I'm not an expert in this space, but I would say that I would predict that most patients are going to show shrinkage in some lesions, growth in others, and transient benefit. I mean, can you imagine a world where we could sample the antigen positivity of each lesion and then create a personalized ADC or vaccine or whatnot, or CAR T that can go after what needs to be done? I don't think you can sample a single lesion and figure out what all the other lesions are doing. I think you need to use some sort of PET scan or ctDNA to get a feel for which lesions are positive for which antigens and develop a combination approach to address these things. So my final slide, my take-home message, sorry I went long, is that the pathophysiology of oncology is complex and it's heterogeneous, and it will not change for us, right? It is not going to suddenly respond to monotherapy because we like developing monotherapies, right? I would argue the monotherapy is a priori inadequate to address complex heterogeneous systems. If your enemy has guns and tanks and planes and satellites and submarines, and all you have is a gun, you're not going to win that war. So I think that our drug development system needs to adapt. We need to find a way to develop combinations and personalized combinations. And that is the only way that you're going to move from curing a small slice of the population to developing therapies that can generate long-term responses in wide swaths of the patients that we're trying to cure. And that's all I have for today. Thank you very much.
Thank you, Brian. We do have some questions. I have one question to start us off. You started this talk talking about modeling and the use of modeling in drug discovery and development. If you apply that modeling to the oncology information you produced, the heterogeneity in the oncology would imply that you'd have to do a virtual population for your model.
Correct.
How then would you define success of the modeling?
Well, we have been using the modeling. We built models of immune therapy, ADC therapy, vaccines, and we create virtual patients that have heterogeneous, density of antigen, density of T cell levels. density of MHC receptors, things like that. And we use these models to make predictions and to look for optimal outcomes. Now, the second stage of every project is optimization. How do we improve this outcome, get more efficacy, less adverse events? And oftentimes the solution is we need to find a combination therapy that's orthogonal to the baseline, and we need to put that in. But finding a way to move combination therapies, especially two novel agents, through the clinic has just been something that it's really difficult to do, right? So I think the oncology space is ripe for mathematical modeling. I think it can generate a lot of insights in this space. We're using it here at Merck. I hope other people are using it out there.
Why do a majority of patients show stable response to pembro in your examples? If the drug works, the tumor should shrink. If it doesn't work, the tumor should grow.
Yeah, it's absolutely fascinating that I thought tumors would like exponential decay. They'd go to 0 or infinity, but they don't actually do that. Especially with immune therapy, we seem to see a lot of stabilization. Target therapy and chemotherapy, the tumors go to 0 or they grow, or they head towards 0, turn around and grow. Immune therapy shows stabilization of lesions. The lesion will shrink by 30% and then stay there for 5 years. It won't shrink anymore and it won't grow anymore. Absolutely fascinating. I don't know why that happens, but it happens all the time. So if you have these lesions that are stable, right, what we generally have for the RECIST criteria is you either get a net growth in the average tumor burden, or you get growth of a target of a non-target lesion, or you get appearance of a new metastatic lesion. And the majority of patients that get described as progressive disease do not have an increase in their total tumor burden. They have a non-target lesion growth, or they have appearance of a new metastatic lesion. Right?
So this question is directed to your rooster.
My rooster.
Correlation. In the rooster analogy, can you please comment on the inverse correlation with respect to the pharma industry? The rooster crowing correlates with the sun rising. Therefore, if the sun doesn't rise, the rooster will not crow.
Yeah, that'd be fantastic. If our objective is to stop the rooster from crowing, and we could find a way to stop the sun from rising, right? So I'm not saying all correlations are wrong. My stats colleagues are going to murder me. They have done incredible work. We found a large number of useful drugs. Statistical correlations followed up by mechanistic modeling to understand the insights generated or potentially not generated by these things. It's just a funny slide to get people's attention and get them thinking about, how much confidence could I put in correlation? Why do we spend $1 million on correlation for every $1 we spend on causation?
We have a couple of questions on cost benefit, and I'm going to sort of merge them. The questions come up about the cost benefit for modeling in drug discovery, and what do you think that is?
Nobody's actually done a study to find out what is the cost benefit analysis of these things. My thoughts on this are, so first off, I think mathematical models work better than thought models, right? And I think that from personal experience, using the model can lead to better decision making. But a lot of these decisions, I wouldn't say don't matter, but they have minimal impact, right? If you find a drug that isn't going to work, you predict it won't work, you get rid of it, and then you replace it in the pipeline with another drug that won't work, what have you done? Right? You haven't done anything, right? The only thing that matters in the pharmaceutical industry is finding a winner, right? The only thing that matters is finding a drug that works. We found pembrolizumab, and it makes billions of dollars a year, right? So finding if you could find one extra drug a year, the economic impact of finding one extra winner a year would be incomprehensible.
Okay, we are out of time. I want to thank Brian for an absolutely fascinating webinar. That was incredibly interesting. And we hope that you can come back sometime and give us another webinar and follow up and see where things have progressed over time. Thank you so much, Brian. And thank you to our audience. Have a great day, everyone.
Thank you.
Bye, and hope to see everybody again at another webinar.
How do I hang?